So much, Rafi. Now it's my pleasure to introduce Professor Caroline Sabin. She's Professor of Medical Statistics and Epidemiology at University College in London. Professor Sabin is the principal investigator of the UK Collaborative HIV Cohort Study, UK CHIC, a major multi-center cohort involving over 30,000 HIV individuals in the United Kingdom. She is also the principal statistician for the DAD study, a large international multi-cohort study of more than 33,000 HIV-infected individuals, and also he has been involved with many other large HIV cohort studies and collaborations, both in the UK and abroad. Her research interests have included include the natural history of HIV infection, the identification of prognostic markers of disease progression, the description of the response to antiretroviral therapy in real-world conditions, and the identification of factors associated with these, and also aging and comorbidities in patients living with HIV. Professor Sabin is a very productive scientist which has authored close to 400 papers indexed in PubMed, and her teaching activity is also outstanding. Professor Caroline Sabin, the floor is yours.
Thank you very much. It's made me realize I need to update my biog to add something a little bit more interesting than work. So I can share with you with Broadway musicals as well, and so maybe we can discuss those tonight. I'd like to thank everybody for inviting me to speak here today and also giving me the opportunity to visit this amazingly beautiful city of Toledo. It's the first time I've been here. And I think it really sort of seems to signify a city that is inclusive and tolerant of people from very different backgrounds and different cultures. And I have to say something that is rather missing from my own country at the moment, in time of political turmoil for all of us. So thank you very much. I also have to apologize for my lack of Spanish. Those of you in the audience who know me know I've tried, but I do have a very good knowledge of anything relating to food and drink, particularly wine. Beyond that, I'm sort of a little bit stuffed. So for the next 20 minutes or so, what I want to do is talk to you about some of the statements that we hear said frequently about HIV and aging. And I really want to question them because I think they've become embedded in our belief system about HIV. And what I want to show is that if we look at some of the evidence in a slightly different way, maybe our interpretation of the data is quite different. So here are my disclosures to start. And just as background, I don't think I need to tell anybody in the audience that the widespread availability of antiretrovirals has really changed the landscape for people living with HIV at the moment. Because of the really dramatic reduction in the mortality rates that we've seen over the last 10 to 15 years, almost all places where we have good access to antiretrovirals are seeing an aging of the population of people with HIV. And the slide here just shows the characteristics of our patients in the UK CHIC study, as you heard about. And you can see that over time, the proportion of those participants that are aging into their 40s, 50s, and 60s and beyond is increasing. And these trends clearly are expected to continue at least out for the next 10 or 15 years. And I suspect everybody has seen the same patterns in their clinic. There's nothing special about the UK. And of course, the problem with that is that as people get older, then people become increasingly susceptible to many of the age-related conditions that you would just generally expect in the general population anyway. So cardiovascular disease, cancers, liver problems, etc. And this slide shows the comorbidity profile. This is from the Age HIV study in the Netherlands, which is led by Peter Rice and his team. And if you look on the right-hand side there, you can see the number of comorbidities that are present in people with HIV as the group gets older. But the real striking thing here is if you then compare that to the pattern on the left, which is the HIV-negative controls in the study, you can see the comorbidity profile for someone with HIV most closely matches that of someone without HIV who's probably 5 to 10 years younger overall. And of course, what this means is the there is increasing concern, not only from the research community, the clinical community, but also the patient community, that HIV is somehow causing premature aging, or that comorbidities occur at an earlier age in people with HIV. And this is almost always the starting statement in any paper that I review or I read. And this is really concerning, particularly for patients who may really worry now that the tremendous benefits that CART has brought with it may now be gradually eroded because of a more rapid rate of progression of aging. And of course, from a research perspective, what this means is our search then continues for biological mechanisms that drive this apparent risk. So we hear many people in the audience, I'm sure, are working in these fields, looking at residual inflammation, or inflammaging as it's known. The gut microbiome is implicated in virtually everything. We have mitochondrial dysfunction and immunosenescence. All of these mechanisms being proposed as explanations for why we're seeing this phenomenon. But what I'm worried about is that in our rush to establish these mechanisms, that we may well have forgotten the basic rules of epidemiology, and particularly now I'm thinking about confounding and the impact that this may have on our findings. Now, why may we see confounding going on here? Well, we know that people with HIV as a population tend to have very different characteristics to the general population, and we know that there are increased risks of sexually transmitted infections in some subgroups, viral co-infections, we've heard about some of those already. There are high rates generally of smoking, recreational drug use, etc. And of course, many of these factors are themselves associated with comorbidities and some of these age-associated diseases. And my question is whether some of these other factors themselves could actually confound the associations that we see. And of course, this is quite possible. This is a nice slide from Jane O'Halloran showing the multifaceted etiologies of cardiovascular disease in particular. And if you look at this figure, you can see that HIV here is only one of many factors that may drive cardiovascular disease in this population. And amongst that, there's other lifestyle factors playing a role as well. So does HIV really cause premature aging, or is it simply a result of unmeasured confounding that we're seeing? So what's being said? Well, the statements are being made regularly. So, for example, people with HIV are increasingly dying of cancer. This is a very common statement. People with HIV develop comorbidities 10 to 15 years younger than their HIV-negative counterparts. HIV is causing accelerated aging. Or it's all due to the residual inflammation or the immunosenescence that's caused by HIV. These are very common phrases. So let me pick these apart one at a time, and let's look at the evidence that supports them. So the first one then, people with HIV are increasingly dying of cancer. So here are some data from the DAD study. Many of you will be familiar with this international cohort. A few years ago, we looked at the causes of death amongst the people who died in the cohort, and you can see, if this works here, this group here, this is the proportion of people dying from AIDS-related causes, and you can see that that proportion is getting smaller, as is the group here, which is a group dying of liver-related causes and potentially the cardiovascular causes there. But the one that is perhaps of most concern is this funny-coloured green bar here, which is the deaths from cancers. And what you can see from that figure is that as a proportion, that is or at least seems to be increasing over time. These are data from the French mortality studies, and these are a national survey of deaths amongst people with HIV in France, conducted every 5 years or so, and this was an analysis conducted from 2000 to 2010. I'm not expecting you to read this slide, but this is all of the different causes of cancer causes of death that they considered in the study. And what they found was that there were significant increases in the proportions of deaths that were from any cancer-related causes, but particularly from respiratory cancers, lung cancers, ear, nose and throat cancers, anal cancers, other hemopathies, and other and unknown cancers. Now, looking at that list, I think you'll probably already start to see that there is a theme for some of those, and certainly some of those have risk factors that may also be separate from HIV, but which may particularly be important in the populations of people affected by HIV. So certainly it looks as if, as a proportion, there is an increasing risk of people dying from cancer, but is this sufficient evidence? Well, if you bear with me with my sort of very simple cartoon here, what I've drawn on this slide is a hypothetical situation where we have a population of people dying from a cause, and in the round circles here, the blue circles are deaths from HIV-related causes, and the green circles are deaths from non-HIV causes. Now, in this hypothetical example here, of the 70 deaths that have occurred, around 57% of those are due to non-HIV causes. Now, if we take away one of those causes because we give treatment and we stop that happening, so in particular if we give antiretroviral therapy and we stop HIV-related deaths happening, we may not stop all of them, but if we stop the majority of them, then what do? Well, we reduce here the denominator, which is the total number of deaths, but in fact the number of non-AIDS deaths hasn't changed at all. And so our proportion dying from non-AIDS causes has now gone up to almost 73%. So all we did here is remove one cause of death. So this is basic maths. If we express as a percentage, then of course if we remove a cause, the proportion of people dying from other causes has to go up to match. So how do we look at this more appropriately? Well, really we should be looking at rates, and these are the age-adjusted mortality rates from the DAD study. And in fact, if you look at rates over time, you can see that for virtually all causes here, this is a downward trend. And this one is the cancers here, so it's potentially a bit harder to see a drop in that, but what you don't see is an increase over time. So if you control for age, it doesn't look as if that's increasing. And what are people dying of? Well, I'm showing some data here from the UK. This is our surveillance data, so not a cohort study, but across the whole country. And on the top there in the darker blue is the death rates from all in the first year after diagnosis. And you can see that for many of those causes, the death rates are very, very high in that first year. But beyond the first year, when you look at the lighter blue column there, you can see that those rates drop. Now, this figure is showing the SMR, and an SMR of 1, so the standardized mortality ratio, would show you the rate compared to somebody in the population of the same age and the same gender. So an SMR of 1 basically essentially shows you that the mortality rates from that cause are the same as what you would expect. And in fact, now if you look at the non-AIDS cancers here, beyond the first year, that's pretty much exactly what we would expect based on the population, their age and gender distribution. So really very little evidence there that there is an increased risk of cancer. So what about the second statement? What about the fact that people develop comorbidities 10 to 15 years younger? Well, this is very, very commonly quoted in the literature. I mean, any paper you read, you can see a statement like this. This is one about lung cancer, suggesting that lung cancer occurs, in this case, maybe 20 to 30 years younger. So is there evidence to support this? Well, this really comes down, I think, to the fact that when you look at the age at diagnosis of a condition, you are essentially conditioning on the age of your population, because you're looking at the people who develop a condition within a cohort which has a certain age distribution. And really to illustrate that, I've just made up another example here. This is a population of people which broadly might reflect the general population. And the red people here are people with the condition. Now, if you look within that population, the average age at diagnosis is 67.5 years. But this population doesn't look like most of our cohorts of people with HIV, because we don't tend to have many people up at this top end here, and we have perhaps a preponderance of people in the younger age groups. So if I change my population, if I get rid of those people who we wouldn't normally see in our cohorts, now what I've done is I've reduced the average age at diagnosis in that population. But all I've done is I've taken the cohort and made it a little bit more what we would expect to see in our cohorts of people with HIV. I haven't changed the risk of the disease at age at all. And yet we've reduced the average age at diagnosis of 10 years, which is exactly what we're seeing in the literature. So how do we get around this? Well, again, the appropriate method to analyze data would be to look at the rates and look at the rates at different ages and see if there is a difference there. And one study that has done this very, very nicely is the VAX virtual cohort study in the US. This is a study from Kerry Althoff and colleagues. Now, the VAX is in a very lucky position that they have a group of people without HIV with very, very similar demographic factors, but also they're drawn from the same underlying population of veterans, so they have the same sort of behaviours, risks that you would expect to see in the people with HIV. And together with an appropriate methodology that the authors used, what they did was they looked at the average age at diagnosis once you controlled for the risk itself. And actually, if you look at the adjusted difference here, there was no significant difference in age at the first myocardial infarction, no difference in age at the first non-AIDS defining cancer. In fact, the only clinical event where they found a difference was for renal disease here, but of course, 0.46 of a year equates to about 5.5 months difference. So the people with HIV did seem to develop renal disease maybe 5 to 6 months earlier, but that's nowhere near the 10 years that we're told about regularly in the literature. And really it is confounding, and this is an extreme form of confounding with age, where you're looking at an association between HIV and a comorbidity here, but you've got age because it is associated with the HIV infection, but also with the comorbidity. And we need to exclude that confounding effect. Okay, so what about HIV is causing accelerated aging? Well, I think to investigate this one, we really have to start to think about what it is we mean by accelerated aging. So just a few diagrams to illustrate this. If we assume that in a population we have an age-associated condition, and what I mean by that is that we have a condition where the risk increases as you get older, and it doesn't have to be a straight line, it's just easier to draw that way. If we have no effect of HIV, then when HIV affects somebody, there is no change in that line. Now, we have a pattern of accelerated aging if we see some challenge, such as HIV, and at that point, the risk with each additional year of age then starts to go up much more rapidly, and that's a model of accelerated aging. The alternative model that we can have is accentuated, where we have our challenge here at this point in time, but beyond that, then the average year-on-year increase is actually the same as in the population without HIV. So those two lines pretty much track each other once you get there. Now, this is not to say that this isn't important, and we need to understand why that's happening, but this is not accelerated, this is accentuated. And then, of course, we can have patterns that combine both of those, so we get some accentuated aging and some accelerated aging. Now, why is this important to me? Well, this is important because ultimately the underlying mechanisms that cause those may be very different. And so we do need to know which pattern it is we're trying to model. The biology is very different if we're looking at something that has a one-off hit on an individual versus something where there is an ongoing challenge as that person continues to age. And those mechanisms will be very, very different. In addition to that, of course, the statistical method that we use to test for each will be very different. And really, if we're looking for accelerated aging here, we have to be able to look at that slope. We have to be able to show that the slopes are going up. And what you'll find in the literature is that studies that claim to demonstrate accelerated aging actually don't provide the evidence to support that. They may show accentuated aging, but not necessarily that there is then an accelerated progress. And it is difficult to differentiate. Ultimately, you need cohort studies for this, or at least longitudinal studies, because cross-sectional studies don't really allow you to look at that slope. So do we have some evidence for this? Well, the next two slides are showing some data from the COBRA study, which is a collaboration between our POPPY study of aging in the UK and the HIV study in the Netherlands that I mentioned earlier. And what we did for this was recruited 150 people from across the two cohorts, and we were able to look over a 2 to 3 year period at a lot of measures of brain aging, particularly neuroimaging results, but also cognition and how that changes over time. We also looked at a lot of biomarkers. I've not got all the data for that, but just to show you today 2 of these slides, these are some of the neuroimaging results. The red lines here are the people with HIV, and the blue lines are the people without HIV. And I think what's quite striking, or particularly struck us here, is that you can see that for most of these, the people with HIV they started off at a worse situation. But if you look at those lines over time, for almost all of these are virtually parallel. So this is really suggesting to us that this is a model of accentuated aging and not accelerated. And we saw the same patterns when we looked at cognitive testing, and we looked at how that changed over time. And in fact, most notably with this is that you don't see really that many signs that cognitive function is getting worse. And if anything, the 2 groups are tracking each other. Again, there is something that has obviously happened at some point in infection, and we need to tease that out. But this is really looking like a model of accentuated rather than accelerated aging. Okay, then finally, what about the residual inflammation? How do we discuss that? Well, again, you'll see this statement made regularly in the literature. People say it's all due to immunosenescence or inflammation. So what did we do with that in POP-B and COBRA? Well, here, this is work from Thijs Bouwman and Neeltje Kutstra, who looked at CD4 and CD8 senescence in groups of people in the red with HIV here. And in the grey colour here, this was a group of blood bank donors from the Netherlands where they had some data. And if you look at that figure, what you can see for all of those is that there is certainly higher values in the people with HIV than in the blood bank donors. And this is true across all of, whether you look at CD4 cells or CD8 cells. And this would immediately think, well, maybe this is something about HIV that is causing this. But what they then did is they went back to the control group that we have in COBRA, which has been drawn from the same underlying population. So these are people, again, largely MSM, attending sexual health clinics. And actually, when they looked in that, if you notice now, if you look at the red and the blue, there are very few differences between the red and the blue lines here, but both groups are very different to the grey, the blood bank donors there. And that was really surprising to us, and we wondered, well, why is that? And we looked at the characteristics of the people. In fact, as the age, because they'd been selected to be age-matched, they're very similar. There was a difference in gender there, but the most striking thing for us was the proportion of people who were CMV positive within the cohort. And this is what we would expect, given the fact that many of the people in the COBRA study were MSM. So what the authors then did was then stratified by CMV status and looked at the results separately in people who were CMV positive and the smaller number who were CMV negative. And when they did that, the differences really disappeared. So now you're seeing much more similar patterns across the 3 groups. I mean, there are still differences there, but they are much less pronounced across them. And what this suggested to us, that these results were not really being driven by HIV, but they were being driven by CMV. So some other factor there that is playing a role. And of course, now we're talking about confounding again, but we have confounding with CMV infection, because we know people with HIV tend to be more likely to have that infection. We know it also drives activation markers. And of course, we thought, well, if it isn't HIV, then maybe it is CMV, but maybe it's something that in itself is associated with that. Again, it's not necessarily a causal relationship. We know that many people, as I've mentioned before, smoke. We have high levels of obesity, low exercise levels, high levels of recreational drug use, alcohol use. Each of those is associated with inflammation. That's well documented in the press. Each of them is associated with mortality and morbidity. And so could it be that actually, rather than the HIV, some of those factors are playing a role? And I'm just going to leave you with one slide here to sort of illustrate the potential for this. And this relates to smoking, and it's one of the factors that has been studied very widely across Europe. We know from the data that's out there that actually smoking now causes more years of life lost in the population of people with HIV than HIV does itself. So maybe a lot of this is being driven by smoking, and maybe the attention should be perhaps less towards the inflammation and more towards dealing with some of those lifestyle factors that perhaps could be either reduced or at least minimized. So I'm just going to leave you that. Just my summary here is that the population of people with HIV is aging. Therefore, there is an increased incidence and spectrum of age-associated comorbidities. We expect to see this. As people die, they will increasingly die of non-AIDS causes, and that is completely to be expected given the age and demographic factors of the population. When we see these statements about HIV and aging, they are often based on a poor interpretation of data or on control groups that are inappropriately chosen. It's not really acceptable to take a control group of your co-workers in the lab because they are not going to have the same lifestyles and behaviors as your population of people with HIV. So it's not surprising that the results may be unclear. Now, there may well be some effect of HIV, and I'm not standing here saying that HIV doesn't do anything at all. There may well be, and I think only time will tell as we're starting to study some of these associations in more detail. But what we feel we're seeing for many of these comorbidities now is a model much more of accentuated aging, and this may be something to do with a historic exposure, maybe to early antiretroviral drugs, maybe to nadir CD4 counts that are very low with people being exposed to immunosuppression. But what we're not seeing really for many of our outcomes is a model of accelerated aging. So really this is good news, because it's good news for us, for clinicians, for researchers, and for patients to know this, that maybe there is not that much of an extra burden of the HIV now on this aging process. So what we really need to do is focus our efforts on understanding the reasons, trying to work out what it is has caused that sort of initial hit. Can we identify appropriate interventions to really limit the harm that is probably being caused by some of those other factors? And particularly, can we modify some of those risk factors in this population? So I'm just going to leave it there. I just want to acknowledge the people who've contributed to discussions about this topic, and thank you very much for listening.